All right. So I think we can start.
Great.
Perfect. With a very colorful title slide here. It's a pleasure to introduce Professor Margaret Gardel. This next talk is about biophysics. Even though Professor Gardel started her studies with studies of physics and mathematics at Brown University, and then she did her graduate studies at Harvard University, completing them with a PhD in physics in 2004. She then moved a little bit away from physics and worked with the cell biologist Claire Waterman at the Scripps Research Institute at the University of California in San Diego. Afterwards, she was a Pappalardo Fellow as postdoc at MIT, and as of 2007, she is the Horace B. Horton Professor of Physics and Molecular Engineering at the University of Chicago, and she's also the director of the James Franck Institute at the same university. Her research focuses on understanding the properties of cellular materials and how they respond to mechanical forces. So the key ingredient here of cellular material is that there's chemical activity, unlike most of the materials that we look at as physicists. Her work also tries to develop new biophysical tools to understand the cell mechanics and even build new cell-like materials scratch. One highlight in her group, they managed to recreate cell division outside of a cell. So her work combines various fields of research, physics, biology, biochemistry, maybe even engineering. And I was also recognized by several awards like the Director's Pioneer Award from the National Institute of Health, the Early Excellence Awards from the American Asthma Foundation, the Inspire Grants from the National Science Foundation, and more recently, the Sackler Prize for biophysics, elucidating the biophysical principles underlying the forces that shape cells and tissues. And so we're looking very much forward to learning about the machinery of living matter.
Great. Thank you very much, Falco, and for the rest of the organizers for putting together this wonderful conference. It's really nice to see a conference highlighting the contributions of women physicists internationally across these different fields. So thank you very much for your efforts to put this together. Yeah, so as Falco mentioned, I'm really interested in developing physics frameworks to understand living machinery. And if we think about the living world, there on the right, the movie playing is actually just a flock of sheep, right? We see this emergence of self-organization and patterns, correlated motions across scales in biology. Right. On the bottom right is a movie of insects taking off, taken with a high-speed camera. So in this case, you see the precise undulations in the wings that allow the insects to rapidly turn and take flight. And then the top right is one of my favorite machines, my son, right, who obviously we all learn how to crawl and control our musculature. Right. So when you think about living machines, you know, at the macroscopic scale, they are characterized by motion, self-organization, dynamics. Right. These systems are all being able to generate forces and respond to forces to be able to drive these dynamics. Now, these examples are all from macroscopic scales that we can observe, you know, in our everyday life. You know, and we want to ask what the origins of these types of behaviors are. And so my lab really focuses on this at the microscopic scale. So a real big advance in trying to understand the origins of what makes living matter different than, say, physical inert matter started really taking off when we could take light microscopy. So this is one of the, you know, Hooke's microscope from, you know, the late 17th century, where you could visualize what the microscopic actions were, right? And so one of the classic examples of where we thought differences between life and non-life were was just a simple experiment where you put pollen grains in water and you watch their motion, right? This chaotic motion, of course, we now know as Brownian motion, right? But at the time, Tom Brown described this as podesis, this leaping of pollen grains, right? And it was thought at the time that this motion was really driven by some animate life forces of these molecules or these pollen grains, that there was something inside the pollen grains that was enabling them to move. Now, of course, it took until the early 20th century when Einstein came up with the theory of Brownian motion that said, hey, we don't need to think about new types of forces of these microscopic scales. All we have to really be concerned with is the fact that there's random thermal fluctuations. These pollens are all in a thermal bath, and these thermal forces exhibit random stochastic stresses onto these pollen grains and drive their Brownian motion. Right, so this is an example where we could make an observation at the microscopic scale. We had a new observation of the dynamics, and yet physics was able to characterize these dynamics from sort of classic thermodynamics and statistical mechanics. With the same type of microscopy, you can start to take movies or images of cells and tissues. So these are two examples that were taken in the modern day with digital microscopy, but could have been done, 150 years ago, albeit at that time people drew images of cells laboriously over minutes and days and hours. And these two examples I have is one is a cell that is leaving your vasculature, so the tissue that controls blood flow through your body. If you do this from a mouse and you open up that vasculature tissue, cells will crawl from this. So this is an individual cell crawling through a soft biopolymer network. And what I like to look at and with this biopolymer network is how much the cell can distort that matrix it's on. So that says as the cell is migrating through this microscale environment, it's generating forces that distort its local environment, right? Of course, in the center is the cell nucleus, where all the DNA information of the cell is stored. But around that is this whole structure in the cell that's allowing this cell to crawl on the matrix. And in the right is a developing frog embryo. A frog, this type of Xenopus frog the frog eggs are very large, so you can actually observe them under very low magnifications. And what you see here is the success of cell divisions, right? So by observing these images, you start to ask, what are the physical models that describe these behaviors that we see of living cells and multicellular systems, right? And so at the time when we observe multiple cells, such as in this developing frog embryo, it was observed at the time from mathematical biologists at the early 20th century, Sir Darcy Thompson, that there's great similarity to the packing of cells, of living cells, with those of soap bubbles. And indeed, this has been a really useful analogy to be able to build models of how the mechanics of cells and of tissues, of multicellular tissue, of thinking of how interfacial stresses really drive the shape of these cells that I'll get into a little bit later. Now, what's really lacking from these types of images of being able to build predictive and models and really engineer these systems is the fact that we didn't know the molecular basis of cells. So an analogy I'll bring up later is at this point, we had no idea what the effective atoms were of these types of materials, right? So over the past, in the 20th century, really, from about 1930s and on, molecular cell biology has become this really rich, powerful field to discover the molecular basis of life. And now these tools are widely used by many labs, and many of the molecules are already known, right? So we can understand what the composition of the materials are that allow cells to do these really exotic mechanical behaviors. And molecular biology, of course, has really served given us a lot of power, because if you want to understand how to control a disease that is regulated at the molecular layer, you can now design targets at the molecular scale. Now, however, from a physical perspective, I just want you to note this big gap in length scales. right? So the molecules that make up cells are at the scale of individual nanometers, right? But the cell behaviors and tissue scales are at the scale of micrometers, right? So now we can't think of cell mechanics and cell morphology as a single molecular event. We have to think about ensembles of molecules and how they work together to build materials and machines that allow cells to regulate their shape. And so just to give you a flavor of what kinds of materials we're talking about when we think about cell mechanics, these are images. Now, instead of with light microscopy as the image of the cell in the middle, these are several different micrographs taken with electron microscopy, where you could resolve the protein-based structures inside the cell. And when you resolve these structures at the nanometer scale, you can start to get a sense that inside the cell, there are these long extended polymers and bundles of polymers inside the cell that are extending and building a scaffolding across the cell that we call the cytoskeleton. So the skeleton of inside the cell, so cytoskeleton. Right, and this cytoskeleton is built from numerous different types of polymers that build mechanical scaffolds that create the cell mechanics. And one of the most important biopolymers is that of actin filaments. And so in this polymer, you have actin monomers that individual monomers are a single protein subunit that self-associate into an elongated polymer that still has a very long aspect ratio. The diameter of this biopolymer is about 7 nanometers, so it's about a little bit less than 2 subunits thick, wide, but the length can be now extended tens of micrometers. Right. And if this biopolymer was a macroscopic material, it would be a very stiff plastic. So we characterize the stiffness of materials as how much energy it requires to deform them by a certain amount. So this Young's modulus is the energy density, the energy per volume, and the energy density of the individual polymer for under compression or extension is 10 to the 9th pascals, which is sort of analogous to a really rigid plastic material. However, because of this long aspect ratio, the weakest and the softest bending mode is, of course, not extension, what you'd expect under extension, it's that under bending. So we have to worry about the bending mode, right? And now the bending mode, the bending energy of the polymer now scales as the Young's modulus times the diameter to the 4th power. And because of this very slender diameter, now the bending modulus is quite soft, and it actually is comparable to thermal energy. So if now we can characterize a length scale known as the persistence length, which is just the length scale of the ratio of the bending modulus, was the energy times length, to thermal energy, and this gives you a length of which tells you over what length scales the correlations of the polymer become uncorrelated. And so this tells you below this length scale, this polymer is semi-rigid. And above this length scale, you can start to think about this as a flexible polymer with no significant correlations in the conformation. And it's notable that this persistence length, if I had put scale bars on these images, the scale bar across this system is around 5 micrometers, right? So this persistence now is why these structures, these single filaments, appear straight under electron microscopy, and it provides some of the mechanical rigidity of the system. Now, so the inside of the cell is coupled by these disordered biopolymer networks, but on top of this disordered biopolymer system, there are distributed mechanoenzymes. These are proteins that generate local mechanical work. They utilize chemical energy to do mechanical work. One of the classic ones is a motor that then binds to the side of the actin filament. This is called a myosin motor. And after binding and utilization of chemical energy known as ATP, it undergoes a conformational change that drives translocation of the actin filament. So the mechanochemical activity of the motor is able to drive the sliding of the actin filament. Likewise, there's other mechanoenzymes that you can even think about the fact that the polymers can be assembled and disassembled as another type of molecular motor, right? So this is an example of the assembly of a microtubule motor, another type of biopolymer, and this is the disassembly. Both of these processes are ways that the biopolymers and the biomaterials can utilize at the protein scale mechanical work to do large scale conformations and generate local stresses. So I'm going to give you one example of a system where we really understand, where, say, the 20th century biophysics really solved the question of how do you transmit stresses to allow motion at macroscopic scales. And this is the example of striated muscle, right? So in skeletal muscle, our voluntary muscle of our body, this tissue has this nice organization across at the millimeter scale. Now, if you look at this tissue, this is a multicellular tissue at the single cellular scale. If you look at a single cardiac cell, you also see a regular pattern along these filaments in the cell. And what's happening in this regular pattern, this is a regular pattern of the motors, those myosin 2 motors and the actin biopolymers, is that along these bundles, their organization and association of the myosin filaments along one part of the actin filament. So all the myosin is accumulated in one region and all the actin filaments are polarity sorted in another region. And because of this organization, each one of these becomes an effective contractile unit that can then take the translocation of the actin-myosin at the nanometer scale and allow contraction to occur at the micrometer scale. And because this now spans at the whole cell scale, this is able to effectively transmit the contractile forces from the myosin 2 motors up to the cellular and tissue scale. So in voluntary muscle, this machine is really regulated by the geometrical organization of the actin-myosin, and allows for this, the amplification and the transmission of the forces across these scales. I will say that, you know, it's still not known how this biological machine self-organizes to build such a really highly organized machine. Another interesting thing to note is, like, once you have built a machine, it's really limited by some of the biochemical function of these molecular motors at this scale, and it's really hard to regulate it through changes in the structure. Now, so this is one example that was really solved before we even had the capabilities to visualize any of these protein actions in live cells. And this was done through, because of this nice organization, we could do biophysical measurements within the absence of dynamic imaging. A really big advance in the field over the past 20 or so years is the feasibility of imaging dynamics of proteins inside live cells and tissues as they're changing shape. And what has enabled this is a few things. From the chemistry side, it's the development of fluorescent proteins that can be, and these are protein structures that can be genetically encoded into living cells, where you shine light of one wavelength and they can emit at a different wavelength. And we have many different types of these proteins, so we can co-localize different proteins at the same time. Coupled with that is the increasing readability of engineering, genetic engineering of living cells. So now we can put these proteins, couple them to something that we would like to visualize in the cell and manipulate the cellular environment. So both of these, the fluorescent proteins has won the Chemistry Nobel Prize, and the gene expression has not yet won the Nobel Prize, but it's a massive tool that's used across biology. And then the third part really comes from physics, is advances in modern light microscopy, so and digital microscopy. So we have tools, many different ways in light microscopy to be able to sculpt our light, to be able to only illuminate a sample where we want it, and eliminate the out of focus light, and very high quality cameras to be able to detect very low levels of intensity. Right, so this is the type of microscopy we do in our lab, called a spinning disk confocal microscopy. And so we load our samples onto the stage, and we are able to visualize through this combination of techniques protein dynamics. And so when we do this, right, what you can see is now, in contrast to the picture I told you for striated muscle, the motion, and what we're visualizing here is both myosin 2 motors in the rainbow scale on the left, and then in green the motors are also in this embryo image on the right. You can see that the systems are highly disorganized. So this is a cell on the left, is a cell migrating up to the right. This is the beginning of Drosophila development, where the motors are generating contractile tension and driving invaginations of the embryo, all these materials, the myosin-driven stresses are highly disorganized, they're changing over time, they're variable with space. So this makes for a much more challenging question in trying to understand, dissect the mechanics of these machines. So I don't have time to go into all the details of this, but many of these machines that we now characterized are highly disordered and dynamic. We know that there's different kinds of machines that exploit the local mechanical forces, local molecular motors to do work, to generate pushing forces, contractile tensions, and also regulate adhesion of cells to their external environment or to each other. And so all these machines are working together to drive morphogenesis of cells. And what I'd like to emphasize is, it's really only been about 50 years since we've been able to build up this molecular scale picture of these machines that are driving cells. And it's been done primarily from the vantage point of molecular cell biology, of identifying the individual components. And I want to make the analogy that, in our studies of physical solids, this is analogous to the idea that, it was several hundred years ago that where we didn't have atomic theory, where we were just indenting physical materials and making measurements and theories of elasticity and we didn't have a picture of what the atomic picture was. So this atomic picture of physical materials has allowed us to engineer them in more precise ways. Yet we could develop Young's law and theories of elasticity without the atomic theory, right? And so the same type of development of physical laws for living matter has still not happened in the same way. And so I want to outline a few challenges of where we've been successful and where we still have in this field. So the first challenge is, as I described first, is that these materials inside cells are highly disordered and soft. Right. So in contrast to that copper metal, right, the interaction between the subunits is on the order of thermal energy now instead of an electron volt, and the length scale is much larger, right? And so because I missed this one part on here, because of the increased length scale, if we think about the energy density, right, that's the stiffness of the material. Because of this large Mike scale, these materials become increasingly soft. Now, in the case of disorder, so this is just a bowl of Jello, so a pile of dead soft matter. In the early 20th century, the idea that you could actually develop physical laws to understand such disordered materials without a crystalline lattice was not clear. And I just want to give by example that we have done this, right? So in the fields of polymer physics, right, we've been able to develop order parameters to be able to describe the disorder in these systems, to be able to make predictions about how their energy density can be changed and how to control their mechanical properties, right? And so this was awarded to de Gennes in the 1991 Nobel Prize in Physics, right? So so this is the one challenge of living materials, but perhaps most interesting in the current day is that we also have to take this disordered scaffold and add all these microscopic sources of activity. And so this is where over the past 2 decades, a new field of physics known as active matter physics has really been created. So when we think of now materials, not most materials that we think of need to have external stresses, external forces applied to them to be able to have a response. But because these biological materials have internal mechanochemical activity, they can constantly deform and drive their own stresses. And so with these are 2 movies generated from my lab of types of active fluids that we can generate from these myosin motors and actin filaments, and an active gel. And we've been able to map out phase spaces over how to control the local stress dipole in these systems and how to also predict the response. Right. And I will say that going back to that first movie I showed you with the flocks of sheeps, right, these theories of active matter are really work across scales. Right. So even the flocks of sheep, you can think of an individual sheep as having a self-propulsion, and there are collective emergence of collective dynamics that occur when you have enough of these self-propelled objects in close proximity. Okay, so we have to think about the activity in there. even less understood than the active matter, right, is really thinking about how these biological materials are changing in response to forces. So their adaptation. And this is a really new area of physics, adaptive materials, where we start to understand how to describe how materials can switch between different types of parameterization. So all of life can be thought of as dynamical systems with sort of time-dependent parameters. So this makes it very different than other types of physical dynamical systems that we usually tackle. And I would say that this is a new field that really has been emerging, of trying to understand how do we understand these modes of adaptive change, not only at the physiological scale in materials like the ones I've been describing, but also like in neuronal networks, but also in evolution of systems, right? So this is something, when we think about dynamical systems in biology, that physics can really contribute. Okay. So when I think about the physics of living matter and exciting opportunities in the future, right, I think about these two different aspects of how do you take these local mechanoenzymes and biomolecules to generate and transmit forces to build different types of active matter. So that's this bottom blue box, but also how is that active material responding to external forces and changing its own physical behavior to adapt and make a new mode of force generation or mechanics. Right. And so together, understanding these different types of mechanical response will really build a picture of how do you build a living machine that can change and drive motility that we see at the cell and tissue scale. Okay, so I only have a few more minutes, so I'm just going to describe one really short project of why I think there's really exciting tools to be able to visualize this now. The example I'm going to give you is in the case of epithelial cells, which is a multicellular tissue. This is an example of a fly wing where I'm going to skip over here, where the fly wing can be thought of as a bunch of, these are now the cell membrane, and all these cells are changing their shape over time and responding to different stresses. So this is a self-propelled flow of the cells that is driving the shape of the fly wing. And we want to develop models of how this shape and this morphogenesis occurs. So this movie, while it's gorgeous, and this is a movie of an actual fly wing development, you can see that there's so many complex flows and forces occurring in it that it's really hard to dissect out as a simple dynamical response in the system. And this is where a new tool that's been developed over the past several years has really allowed us to control the dynamics, the biological dynamics. And this is a tool known as optogenetics. So this is a tool that, again, comes from chemistry, where there are light sensitive interactions between molecules that can be genetically encoded inside the cells. So when we shine light on a portion of a cell that contains these two components, it drives a localization of these components to change the activity. So this allows us to control where forces are being applied in the system with our laser beam. And so this is just an example where we're shining light in the upper left of the cell, and what we're visualizing here is the recruitment of this protein. So this just shows that we can do this in a reversible fashion. And this allows us to measure response functions in this dynamic material. So the example I want to show here is where we've done this in highly packed suspensions of cells, and we're only shining light on this one interface between two cells. And when we shine light here, we're increasing the amount of contractile tension at this interface. So we're driving contraction that's in response to light, and so the interface is shortening because there's more tension at that interface. And if we turn off the light, the interface goes back to its original length. So this is just analogous to the idea of a spring, right, where you're increasing the contractile force, and then you're taking off the increased force, and the spring goes back to its original length. So this is exactly what we would expect for like a physical system where there's no modifications. But we were interested because in cells you see the length of these interfaces change over time. What would happen if we increase the length of the interface further? And what we see is that if we increase it long enough, we see it becomes switched to being non-reversible. And so now we increase the duration of that contractile pulse, the interface contracts. now there's some remodeling that occurs, and then the interface becomes now at a permanently shortened length. And so we can now map out this response through application of different stimuli and see what's the time scale and the amplitude of the stimulus that's required to drive a irreversible shortening. And so this shows us that for short interfaces, it's completely reversible, but then there's this rapid switch at longer timescales where it seems to become irreversible. And this allows us to now build dynamical models of this irreversible behavior and this adaptive change that's occurring at these junctions. And so the type of adaptive change that we say is happening is that there's a thresholded response to the amount of tension and how the tension is changing in the junction. And so this was theory work done by our collaborators, our theoretical collaborators at Carnegie Mellon, Sheila Banerjee and a student, Mike Stadden, to visualize the response that's occurring. And so this model that we could then test, and this is the beauty of having this tool of optogenetics, is that we can go in and now probe the dynamical response experimentally and test it with these theoretical models. Okay. And the one example I want to give of what you can learn when you have these dynamical models is the fact that we can now ask a thought experiment where we could say, like, okay, we saw this irreversible shortening with one long pulse duration. If we apply the same stimulus, the same integrated stimulus, what happens if we separate that stimulus into two shorter pulses? And what we can see is that, you know, when the pulse is when there's one long pulse, the system saturates and there's no further contraction, right? The whole junction just contracts by 20%. But as we separate that pulse into two shorter pulses, you're able to, after you go through this refractory period, you're able to drive a further remodeling event to drive further shortening of the junction. So this is predicted in the theory. And then we were able to test that in the experiment to show that by this spatiotemporal sculpting of the stimulus, we could actually change the different types of morphogenesis that we can build in the system. Okay. What we think is happening at the molecular scale is that these local stresses are driving endocytosis of the materials that are, so internalization of the materials, the adhesion between the cells. So there's a local remodeling event that's occurring, and we can manipulate that remodeling and show that we can't get any shortening of the system. And I guess what I would like to end with is this idea that, like, you know, the question we have is, like, what are the origins of the adaptive change? And with these tools, we can now probe the length scales over where you're seeing adaptive remodeling. And in this system, we can see that that's happening at the scale of a few microns. So it's way bigger than a single molecule. It's smaller than the cellular scale. And it keeps occurring. This sort of scale of adaptive change happening at this micrometer scale is something that we've seen quite often. And so we're really interested in exploring what controls that and how to manipulate that kind of adaptation. And so for the second part, you know, I've just given you one insight of the kinds of experiments we can do in this field, where we have tools of imaging and physical manipulation to probe dynamic responses of cell dynamics. These new tools are giving us really good experimental control of probing mechanics of living cells and tissues, and really test physical models in a way that we couldn't before. And it's giving us insight into the kinds of force-activated behaviors that are underlying the mechanical response of the systems. And I'll just end with this really sort of forward-thinking idea where I think the field is going, is that, you know, in physical machines, right, we had to have this coupling between developing physical laws and then optimizing the types of physical machines we could build, right? So there's always this interplay between building up predictive physical theories, maybe being inspired by some industrial need, but that allows for further engineering and optimization, right? In the same way, we have not built those laws of physics for living machines, right? So we still need the tools of building active and adaptive materials and matter from a physics perspective, so that we can get to a point of engineering these living systems, so we can actually control the morphogenesis of cells and tissues. And this will really allow us to have a new tool to basically for tissue engineering to control the shape and form of tissues, but also develop new types of robotics that require these laws of physics. And then I just like to thank my group. I have a group that, as was mentioned before, is highly interdisciplinary between people coming from physics, cell biology, and engineering. And as with the previous speaker, I'd be remiss not to use this opportunity to encourage, if you're interested in working in this area, come join us in Chicago. And we always love to work with new people. So thank you very much.
Thank you very much for this great insight into new materials and a new type of doing physics, basically. There are questions. Tamara has a question. Do you want to ask it yourself?
Yeah, I can do it. I can also just maybe read it out, because my question was whether the research results in your field can help in medicine, for example, advancing I don't know how to pronounce that word, prostheses, because you will be able to better understand the organization and the dynamics of the tissue.
Yeah, I mean, that sort of like speaks to like the last slide, is that I would say that currently, I mean, first of all, it's amazing what can be done without actually having control. That basically you can meld this interface between living and non-living materials can work. And yes, a hope I would have would be that this would allow you to build, like, so the big field right now is how to improve our ability to create organs outside from say stem cells, right? So you don't have to do experiments in mice and other animals, right?
Yeah.
So even this idea of how to engineer organoids and how to engineer systems that build outside of cells is something that I really think is an exciting area for the future. But also how to improve surgical techniques, interfaces at the living and non-living interface, I think would be very exciting opportunities.
That sounds really exciting. Thank you.
Okay, maybe I also have a question. You stressed the point that these materials are very disordered, or these kind of systems are very disordered and soft. And so is there, I mean, in the end, things still seem to work out, right? It's not that every member of a species works differently. They all still kind of work the same. And so in the end, the question is kind of, there needs to be some sort of universality that perhaps could help you in modeling these, just like there's universality in solids, even though solids are also could be disordered, but there are certain phenomena that are universal, right? And is there some degree of that one can use in modeling and the description of these kind of systems?
Yeah, no, that's a great question. I think one of the challenges that I think that we have, especially with making predictions of the dynamic system, is this last part of really how the physical parameters are changing over time. So I have been, like coarse grain theories are really good, right? And they work, once you have them described, they work really well, but you do need to know like on what scale you can coarse grain and how the parameters of are changing over time. And I think those are things that in contrast, to physical systems, those length scales are changing, and sometimes right, and for different systems it might require a different type of coarse graining, but also which parameters you put in there and what their rules are for changing is also challenged. So those are, I agree with you that our hope is that there would be generalizable theories. I'm always a little bit careful to be overambitious about that, but I do think that there is hope. If we can have more experimental handles on doing the manipulations, in the same way that condensed matter physics has, right?
Yeah, I mean, I think what you showed in the end is a great start, right? If you're able to manipulate these things at like microscopic or macroscopic level and over different scales and learn the response of that, I think that sounds
that's
like
right. Yeah, and like
yes.
I think building the system, because the field, I mean, if we just think of condensed matter physics as a starting point of the success of how do we think about collective phenomena in large collections of objects, right? Even figuring out how to design those experiments for biological and living materials is something that I think our field has had to sort of grapple with, and I think it's definitely getting there because of both advances and also just building a field that is able to have that kind of communication.
I see. Okay, so Jose has a question.
Yes, I have a question. So thank you very much for this really very nice talk. My question regards your optical measurements. So in order, for instance, this example that you show us, that you are able to change basically in these walls the length, which is the energies of your laser? So at which energies are you having these effects?
Yeah, so I mean the laser light effects, I mean, you know, the lasers are very low. There's no heating going. I mean, there's very minimal heating going on, right? So the energy is not coming from the laser. The energy is coming from this light-induced conformational change to the protein scale. Does that make sense? I guess the energy is coming from the laser, that on the molecular scale. But what's happening is at the molecular scale, there's a change in binding affinity between two proteins, and that's then recruiting chemical species to then do the work. Does that make sense?
Oh yes, a lot.
Yeah. Okay. So yeah. Yeah. Because if we heat up our sample, the whole cell just goes. It doesn't like it that much.
That's why I was asking exactly.
Yeah. So these are constructs that have been engineered. They're from photosynthetic constructs, light sensitive moieties that then have been optimized for light sensitive interactions. So yeah, the most of the energy is coming from that local chemical light induced changes in chemistry.
Okay, thank you very much.
Okay. Laura, do you still want to ask your question?
It was actually not my question, but it was a question by the audience. Let me see in the chat. Yeah, so the person asked, thanks so much for the very interesting talk. Could you please explain again how optic genetics works?
Oh, yeah, so the optogenetics is, so what happens is we use genetic engineering to express two new proteins into the cell that it doesn't know about, okay? And these two proteins have two components. Let me go back over to this image, actually. It'll be easier. Okay, so one of these components, so there's, first of all, these two proteins are here because the blue one, this love domain, when you shine light on it, the light unravels this J alpha, this component, this little tail of the protein, and it's an alpha helix, and then that alpha helix has a high affinity interaction with this PDZ domain over here. And so in the presence of light, there's a high affinity interaction between these two proteins. So that's just the light sensitive interaction between the proteins. Now, in addition to those two, that light sensitive interaction, one of these protein components is bound to a place on the cell that you would like to shine light on. So in this case, we put it on the plasma membrane. So we put a little part that allows that protein to be always sequestered to the edge of the cell. And then the rest is just diffusing around inside the cell. And so that also contains the protein we'd like to concentrate in that region. So in the absence of light, that red protein is diffusing around in the cell. In the presence of light, that red protein gets concentrated on the cell membrane. And in the case that I showed you, that red protein, that red bit is what's generating extra contractile force. So that's extra chemistry. So we're just like hijacking the concentration of that chemical species to do other kinds of work. Okay.
Perfect. Thank you so much.